In order to be used for long-term energy, as we described in the first part of this chapter, we also need to be able to make fatty acids and lipids to store energy or to fill other roles. The remainder of this chapter will be talking about how acetyl-CoA can be used to build up fatty acids, a process called lipogenesis, and also how acetyl-CoA can be used to build up cholesterol, and then thinking about how all these pathways, both the catabolic and anabolic, fit together with our hormonal control. Lipogenesis, which again is the anabolic pathway where we synthesize fatty acids from acetyl-CoA, is very similar to the reverse catabolic counterpart, fatty acid degradation. Because we sometimes need to synthesize different fatty acids than are available, both processes will sometimes occur at the same time. For this reason, they need to be separated and controlled individually. To accomplish this, our bodies will perform lipogenesis in the cytosol, physically separating it from the fatty acid degradation that happens in the mitochondria. It also uses different enzymes that can be controlled separately, and a different carrier molecule for the fatty acid and the carbon source. We also spend energy in a very particular way. NADPH is the main source of energy as we build up a fatty acid. All these differences are important to maintain independent control over these mostly opposite pathways. Because we usually generate acetyl-CoA in the mitochondria, whether it's produced from pyruvate or from fatty acids, the first thing that we need to do to synthesize new fatty acids is to move it out into the cytosol. This is the purpose of the citrate-malate pathway. Citrate and malate can both move in and out of the mitochondria fairly easily with specific transporters. They're also both components of the citric acid cycle. Inside the mitochondria, we essentially run steps 8 and 1 of the citric acid cycle to convert malate to citrate. Notice that it produces an NADH in this process and takes an acetyl-CoA and moves it onto oxaloacetate to make citrate. The citrate can move into the cytosol, which we see on the bottom of the figure, and in the cytosol, we'll spin an ATP to move the acetyl-CoA back off of citrate and make oxaloacetate, essentially running step 1 of the citric acid cycle in reverse. Then we spin an additional NADH to run step 8 in reverse. While it seems like we might be losing energy, this actually gives us a way to move NADH electrons in the cytosol from glycolysis into the mitochondria, so it's essentially a wash. Acetyl-CoA is the initial source of all the carbons in lipogenesis, but we need to move them onto a carrier protein that will be able to be recognized by the lipogenesis enzymes. You can see this step in the top row, essentially just swapping out the acetyl group from a CoA onto the acyl carrier protein called ACP, which literally is an abbreviation for acyl carrier protein. This is just for the first 2 carbons. Everything else needs to be added to this in 2 carbon sections, kind of the opposite of beta oxidation. We also need to activate the 2 carbon piece we're going to add on, because it's harder to add carbons together than it is to break them apart. We do this with an acetyl-CoA by capturing a carbon dioxide, which costs an ATP. You can see this in the second row. This carbon source also needs to be moved onto an acyl carrier protein so that lipogenesis enzymes can recognize it as well. So the malonyl that's produced on a CoA gets moved onto an acyl carrier protein also. This resulting malonyl ACP, which we can see on the bottom, is the source of every 2 carbon addition to the chain after the first and represents an investment of an acetyl-CoA and also the 1 ATP used to activate it. Starting from an acyl group and increasing by 2 carbons is a process that happens repeatedly in lipogenesis, just like beta oxidation repeated over and over again in fatty acid degradation. The first step, which you can see on the top of the left side, joins the main acyl group with the activated carbon source, malonyl ACP. Remember from the last slide that malonyl ACP was already worth an acetyl-CoA and an ATP, essentially 11 ATP altogether. The resulting 1,3 ketone, the 1,3 just referring to the relative positioning, goes through the opposite process that we saw in beta oxidation. In this case, a reduction to an alcohol with NADPH, dehydration to an alkene, and then reduction of the alkene to an alkane, in this case, again using NADPH. At the end of this process, we have an acyl group that's 2 longer than it was before, and we spent the malonyl ACP worth 11 ATPs, and also 2 NADPHs, which are the equivalent of 5 ATP. Each cycle of lipogenesis is then worth 11 plus 5, or 16 ATP. Note that this is more than the energy we get out of beta oxidation, which was only 14 ATP. Just like with glycolysis versus gluconeogenesis, the anabolic pathway costs more than the catabolic pathway generates. Nothing is free in life. Again, this set of 4 steps will repeat for every 2 carbons that are added until the chain reaches the desired length. The first 2 carbons don't need to be activated or reduced, so they just cost 1 acetyl-CoA. But every set of 2 additional carbons costs an acetyl-CoA, an ATP to activate it, and 2 NADPHs for the reduction, an equivalent of 16 ATPs. So for the 16 carbon fatty acid shown, we'll have a number of cycles equal to 16 carbons divided by 2, since the carbons are added 2 at a time, minus 1, since the acetyl ACP isn't cycled, it's just transferred from acetyl-CoA. 16 divided by 2 minus 1 equals 7 cycles. To figure out how much energy was spent to make this molecule, we'll say that 7 cycles times 16 ATP per cycle plus another 10 ATP for the acetyl ACP that isn't part of a cycle. So 7 times 16 plus 10 is 122 ATP in order to produce a 16 carbon fatty acid. We can't make unsaturation at all locations on a fatty acid, but we can make some unsaturation in our bodies. We accomplish this by using oxygen to oxidize both an alkane to an alkene and NADPH to NADP+. This means that it actually costs us energy to oxidize an alkene, where in all other oxidations we gained energy. We consider NADPH essentially like NADH, so again it's worth 2.5 ATP. Each double bond then costs us an additional 2.5 ATP to make. So when we calculated the yield of 16 carbon fatty acid last slide, we found it cost 122 ATP to make. If we had a 16 carbon fatty acid that also had one double bond, 16:1, it would cost 122 plus 2.5 ATP, or 124.5 ATP. If we had a 16 carbon fatty acid that also had 2 double bonds, it would cost 122 plus 5 ATP, or 127 ATP. Acetyl-CoA isn't just used for synthesizing fatty acids. It can also be used to make cholesterol. Your body not only makes the cholesterol, but it also makes the lipoproteins that carry it, either a high-density lipoprotein, HDL, or a low-density lipoprotein, LDL. The majority of your cholesterol comes from your own biosynthesis, not actually from your diet. And even when it does come from your diet, in both cases, your body decides which carrier to give it. So ingesting cholesterol really isn't what gives us cholesterol problems. High LDL, that's again the low-density lipoprotein that transfers cholesterol out into the tissues, that has a lot more to do with eating trans fat. Remember, trans fat usually comes from hydrogenated or partially hydrogenated oils, whether or not it's shown up on a nutrition fact label. Smoking can also lower the HDL levels. That's generally the good cholesterol. So, don't. Regular exercise can improve HDL levels, particularly if people hit certain thresholds of activity, typically starting at about an hour of moderate intensity exercise a week. You might not realize this, but Diplodocuses also metabolize fatty acids. And to tell you a little bit more about that, I have a younger Professor Bosher, Professor Levi, to explain.
Diplodocus name means double bean.
Is there anything else you wanted to tell them about?
And it lived during the Jurassic, and it aged, and it didn't survive into the late Cretaceous, and what else could I tell them?
Maybe what type of dinosaur it is.
A sauropod?
Yeah, thanks, Professor Levi. It's always helpful to see pathways all together to see how they're connected. Fatty acid degradation and glycolysis are both routes to supply energy aerobically. Fat is a much better way of storing energy, and our bodies can store a lot more of it than glucose or glycogen, so we're always going to use glucose preferentially first. With this in mind, we're going to run through a few scenarios of what might be happening in our bodies. The first is that we have just eaten a big meal. If we have just eaten, we will start to take up monosaccharides, fatty acids, and amino acids in pretty large amounts. That means that glucose levels are going to surge in our bodies, and our bodies will release insulin. Insulin will promote using up all that extra glucose. In order to do that, we'll increase the rate of glycolysis to the point where it's supplying the majority of our ATP. And since we only need so much ATP, we're also going to decrease fatty acid degradation at the same time. This means that most of the fats will be stored for later in adipose tissues, so fatty acid catabolism will be fairly low. If we still have really high glucose as we meet all of our ATP needs, which is actually pretty likely, our bodies will also make glycogen to store it for later, and we'll also use the pentose phosphate pathway to power lipogenesis as well. Ketone bodies won't be produced since there's plenty of glucose. Gluconeogenesis will also not be active unless exercise is occurring at the same time. Okay, for a second scenario, let's say someone is sleeping. They haven't eaten for a while, so their blood glucose level is going to drop. That triggers the release of the hormone glucagon. Glycogenolysis may occur to supply some additional glucose, and the majority of ATP production is going to shift away from glycolysis to fats and be produced through fatty acid degradation. Because these can't supply the brain with energy, they will also be used to perform, the acetyl-CoAs from fatty acid degradation will also be used to perform ketogenesis and increase the ketone body concentration. That means somebody who is sleeping will have a decreased blood pH to some extent. Very little lipogenesis is likely to occur since there isn't a lot of extra energy. Gluconeogenesis also probably won't occur either since we can't make glucose from acetyl-CoA and there's no new source of pyruvate or lactate in order for us to synthesize glucose from. Okay, so now our third scenario is someone who's on a keto diet where they primarily consume fats while also limiting carbohydrates and protein intake. Since glucose levels will remain low, glucagon remains the dominant hormone, and most of the ATP produced will come from fatty acid degradation. This will look a lot like fasting in the last scenario. Depending on how long the carbohydrates stay low, though, and how low they go, this can be pretty dangerous. Ketosis will decrease the blood pH and makes it a lot harder to breathe. The longer you're supplying glucose like energy through the ketone bodies, and the higher those ketone body levels get, the more problems one is likely to see. In the next chapter, we're also going to talk about how the metabolism of amino acids from proteins fits into this larger metabolic picture as well.